Good evening, everybody. Just going to give a little bit of time for people to get into the room. All right. Well, for those of you who don't know me, I'm Reg Hoyt. I'm the co-chair of Animal Biotechnology and Conservation Department, and it's my honor to be the chair of Del Val's One Health Working Group as well. And I'd like to welcome you all to this semester's first One Health seminar. I imagine that a vast majority of you have had some exposure to One Health already at Del Val, but for our freshmen, let me just kind of give you a quick rundown on what One Health is, and why we're so interested in this particular area. Well, to make it very simple, we got one planet, uh, and we're all living on this planet together with everything else that's living on this planet. And we're engaging in activities related to the environment, whether it was the biological side of it or whether it's the physical side of it. And we have impacts on each other. It can be good or it can be bad. For us to be able to, no matter what major we're in, consider what our impacts are in the world, it's a pretty good way to work. And it's our belief that we should approach things as a systems approach, and that problems, whether they're here regionally, whether they're regionally or nationally or internationally or global, uh, that it really takes multiple disciplines to deal with these problems. And we have a number of courses that actually deal with this and kind of point out to you why it's important to kind of listen to others, and take some advice from other folks. The way we are approaching this is that we do have a lot of courses at Del Val that are related to One Health. They may not necessarily say it in their title, but they certainly are going in that direction with it. We do have a One Health Communications minor, and we just started a brand new One Health Masters of Public Health, for those of you who may be thinking about going on to grad program. Also encouraging our students, and the one thing I love about Del Val is that student research clearly is student research. And we'd love to see a couple of you get together, or maybe three of you get together and come up with a nice One Health project to be working on. It'd be great to work across departments. We don't have much in the way of silos, and it'd be nice if we tore down what little ones we do have. And then the other part of it is outreach. And actually, this seminar series is part of that outreach. It's our goal to actually open this up to everybody. So there are probably many people on this that are actually uh, members of our community. We welcome you, and we hope that you tell your friends about this and that you come back for other presentations. One thing you should note, and you may tell others too, is that we record all of our seminars, and these are stored at delval.edu/onehealth, one word. So you can come back, and if you think you missed something or you want to share it with a friend, you can do that. At this point, I'd like to introduce our guest this evening, and we are incredibly lucky. We have a fabulous speaker with us this evening. Uh, he was approached because I had read his book, and found it to be incredibly entertaining as well as incredibly informative. And I hope that after you listen to his presentation, you're going to go out and get his book as well. So tonight we're very happy to welcome Dr. Arturo Casadevall. He is an MD, a PhD, is a Bloomberg Distinguished Professor and Chair of the Molecular Microbiology and Immunology at Johns Hopkins School of Public Health, received his MD and PhD from NYU, and his residency in internal medicine at Bellevue Hospital. He's the author of over 1,000, you heard right, over 1,000 papers. His major research interests are fungal pathogenesis and mechanisms of antibody action. He was a leader in the deployment of plasma during COVID-19. Thank you very much for that. His interest in scientific enterprise issues and with collaborative showed misconduct accounts for the majority of retracted publications. He is editor-in-chief of M Biology, deputy editor of the Journal of Clinical Investigation, and serves on numerous editorial boards. He has served on several NIH committees, NIAID Strategic Plan, Blue Ribbon Panel on Biodefense Research, NAS Panel, our Federal Regulations and Reporting, and National Science Advisory Board for Biosecurity. He was commissioner of National Commission on Forensic Science and previously serves as president of Medical Mycology Society of the Americas. He served as the chair, board of governor for AAM, and has received numerous honors, election to ASCI, AAP, AAM, fellow of the AAAS, NAM, NAS, distinguished fellow in immunology and CFR. Dr. Casadevall has authored 2 books dealing with scientific issues. In one, What If Fungi Wins, which we'll be talking about this evening, introduces readers to fungal world, discussing all of its benefits and threats. And in Thinking About Science, Good Science, Bad Science, in How to Make It Better, written with Dr. Ferric Fang, he addresses current science while discussing the problems of the discipline. Again, thank you so much for being with us. I hope you'll all enjoy his presentation.
Thank you so much, Reg. Thank you for having me. And let's see if we can share. And it looks, is this working? Yep. So
yep, you're good.
This talk is already a success.
Sorry, if folks have questions, please put those into the Q&A. And at the end, we will come back to those and hopefully we'll have time to get those answered. Thank you.
Thank you. So my goal here is to basically have some fun while introducing you to the wonderful world of the fungi. So what we're going to do is we're going to hop around on a variety of stories. So, oops, what is in? There we go. We're going to talk about the world's largest organisms or closest relatives, a cold kingdom. We're going to talk about melanins and fungi and the origin of mammals and global warming. So you can see it's a diverse group of subjects. We're going to touch on them, but the idea is to give you a sense of the wonder of this group of organisms. So let's start. The world's largest organism is a fungus, and it lives in the Oregon forest. It's very old. It's between 2,000 and 8,000 years old, and it weighs as much as a World War II battleship. It causes a disease in plants, and the way in which you can tell the organism is because the plants on top of it begin to die, and if you take out the mushrooms and you bring them to the lab and you do DNA analysis, you can see that all around the perimeter of that thing is the same organism, and yet you don't see it because it's under your feet. The only time you see it is when the mushrooms come out. It turns out that when you walk on the forest and you see a mushroom and you look at the plants, the mushrooms are closer to you than the plants are. And this was a revelation about 30 years ago that is actually very medically important, as I'm about to tell you. So somehow about more than 1 billion years ago, it looks like the animals and the fungi split. But the fungi retain a lot of the biochemistry that we have. And that is a problem when it comes to antifungal drug discovery. Because what you want to do is you want to find a difference. But if you're so close, it's very hard to make antifungal drugs. And consequently, we have very few of them. It's a cold kingdom. These were pictures taken by Radamés Cordero, my colleague, during the COVID pandemic. He went out for walks in the woods, and he took we had a thermal camera that we were using to study melanin. I'll be telling you a little bit about that. And he began taking pictures, and he noted that the mushrooms were always blue. Everything else had other colors. And the reason for that is the mushrooms were cooler than the environment. How much cooler? Pretty cold, 5 to 6 degrees centigrade. That's a big thermal difference. And what we went into the literature and found out that people have reported this. So what we contributed is we asked the question, what happens to the microscopic fungi? Does this carry it over to colonies of fungi? And what you see there is a lot of colonies, and they're colder than the environment. But the interesting thing, look at the right. What you see in there, in each of those graphs, is a thermal graph across a colony. And what you can see is that as you get to the edge of the colony, the temperature goes up because the fungus is putting out enzymes to digest the nutrients, and then it drops rapidly. So even in the microscopic world, the fungi are trying to stay colder than the environment. If you put the fungi at 4 degrees, they try to be colder than 4 degrees. And they do this by transudating, the same mechanism that makes you cold after the shower. And in fact, as long as they have water, they stay cold. And we were able to create an air conditioner we called a microcooler, in which we demonstrated that just with mushrooms from the supermarket, you could cool things by about 10 degrees centigrade, which is significant. Fungi are being used in many other ways. It turns out that you can make fungal leather. It feels just like leather. Companies are developing this. This avoids the use of animals, and you can get the fungi to grow, the mycelia to grow in a shape that you like. I'm going to tell you about melanin, which is a fascinating pigment. It comes from the Greek word melan, or black. They're usually dark black to brown. It is found in all biological kingdoms. Everything makes melanin. The protozoa, the plants, the fungi, and we do, and certainly we have it in our skin. It protects us against the sun. But it's associated with a lot of functions. But what I'm very interested in is very resistant to strong acids. In fact, before I go there, this is the world of melanins. The ink from a cuttlefish are melanin particles. These colors that you see there are melanins. Here you have a banana on the right trying to repair itself by making a melanin patch. And look at these are ducks. If you go to ducks anywhere, they're all black fungal things. So melanins are everywhere, and they are multifunctional pigments. This is what our fungus looks like. When you grow it, you give it a substrate, in this case, L-DOPA, that allows it to become completely melanized. The melanin is synthesized in specific organelles, just like in your skin, in melanosomes. And it's made intracellular, and it migrates to the cell wall, and it sits on the cell wall. And why is it important that it's in the cell wall? Because that is the part that faces the outside. So the melanin is put in a position to protect the fungus against outside injuries. Now, if you take those black fungi that I showed you and you boil them in hydrochloric acid, you can recover what we call melanin ghosts. Everything is digested. The DNA, the RNA, the lipids, everything goes. But you're left with a melanin shell. If you take non-melanized organisms, they completely go into solution. This stuff is the only thing I know in biology that will resist boiling in concentrated hydrochloric acid. It is a remarkable, strong substance. And when you look at these things, what you can see is this is atomic force microscopy, that they are composed of even smaller pieces. The fundamental unit of melanin is a very small piece. And I like to think of it as something as an armor. It's not a full plate because it allows diffusion. It's not chainmail. It's somewhere between studded leather and chainmail. So over the years, we have shown that melanin protects against practically anything you throw at it. Ultraviolet light, heat, enzymes, your favorite oxidants, defensins, which are small peptides, heavy metals, and it protects against antifungal drugs. So this is a mechanism by which these fungi can resist antifungal drugs. They put on melanin and become more resistant. And here is a wild story. This is the Chernobyl disaster. So this was a chemical explosion that happened in the old Soviet Union in 1986, in the where is now the Ukraine, and the fungus, the reactor melted, and they created a sarcophagus around it because they needed to keep the other reactors working. And around the turn of the century, it looked like it was going to collapse. And the European Union sent in that robot there. And you see the walls are black. And they're black not because of a fire. They're black because they coated with black fungi. In fact, black fungi are thriving in the high radiation environment of the reactor. If you walked into that area, you were lethally radiated in about 5 minutes. So we initially thought that maybe melanin was protecting against radiation. And the answer is yes, it is. But that couldn't explain why that. Why were these fungi attracted to that very high radiation environment? It turns out the fungi are naturally very reactive, so resistant to radiation. So this could not be the explanation. But even so, melanin has about half the shielding capacity as lead. So it's a biological shielding pigment. So in fact, we have used it to show that if you feed a mouse black mushrooms, the ones that you buy in the store, you can protect the mouse against lethal radiation. And the survival, this is published. So how does this work? Well, the mouse is still getting radiated and its bone marrow destroyed, but the melanin protects the gut. And by protecting the gut, doesn't allow the leakage of bacteria. And the other thing that happens is the gut then reseeds the immune system. So we published this, and it's been confirmed by another group. And there's a lot of interest from the use of melanin as a rapid protection against a radiation problem. Now, melanized fungi, and this is the clue that made us think that something else was going on, tend to migrate to our radiation source. So if you put a radiation source, the fungi are attracted to it. Why would they grow towards a radiation source? And we began to put two and two together and said, well, maybe something is happening, that they're using the radiation for food. And that is what we proposed. We found in 2007. In fact, we worked out that the melanin could capture gamma rays. Yes, gamma rays, highly energetic. And in fact, the melanin that triggers the reduction of NAD to NADH in biological useful energy. So when we had this finding, we were very excited, and we sent it to a one-word journal. You can think of what word journal that was. And they had it for a year. They made us do more work, and they rejected it. And then we sent it to a second one-word journal, and they again rejected it. We had 3 reviews. One review said they should publish this because even if they're wrong, this is going to stimulate a lot of work. The second reviewer was part of the mechanism police and said, you shouldn't publish this unless you can trace the electron all the way from melanin to ATP. I said, you can't do that with photosynthesis. I mean, she shouldn't publish photosynthesis. But reviewer 3 is the one that did us in. The reviewer said, I can find nothing wrong with his paper, but I don't believe it. So at that point, they rejected it. We went to +1. They gave us additional work. And this is what happened afterwards. The journals that rejected our paper ended up writing stories about it. And you can guess what the one-word journals were. But in the end, the data is out there, and it's a wonderful story. There was one thing that bothered me. Somebody said to me, Arturo, X-rays go through things. You're telling me that gamma rays that are even more energetic are being stopped by melanin. And for a minute I said, oh my God, you're right. We must have made an error. And we spent 4 years just trying to show that melanin could interact with gamma rays. And this individual here, Charles Turek, did the critical experiment. He made a melanin electrode. He put then he put the electrode under a gamma beam, and he got a current. And the electrode without the melanin did not produce a current. So that, I think, tells you that in some ways, the melanin is able to interact with gamma rays. And this has been now confirmed by other groups. This is important for astrobiology. Fungi are overrunning our space station. And they attack the door, they thrive in the high radiation environment of space. And this is a continuing problem, and this will be a huge problem as we begin to build larger structures in space. NASA is very interested, however, in the possibility of using fungi for the Mars project and returning to the moon and things like that. And they call it mycotexture, NASA's mycotexture initiative. And there's a link, and you can read up on it. The idea is these organisms can resist the vacuum of space. They can do a lot of things. And people are thinking in terms of future biological construction materials. The melanin in the fungi, it can be found in lichens. So the Black Mountains of Antarctica, composed of a lichen and an alga. And those that we think that Black Mountains, they're using the melanin as a primitive form of energy capture. However, when these glaciers begin to melt, holes appear and they're colonized with Black organisms, and that only drives it up because the water gets progressively warmer. So melanin is involved in many aspects of the biosphere. I'm going to introduce you to a new concept, disaster microbiology. And that is the idea that when a disaster occurs, all the microbiomes change. You see a tsunami, you see the destruction. But when the waters recede, the microbiology has been changed. And this is a new idea that we need to begin to take on, because the changes in the microbes can also affect the recovery. It can affect many other things. And we are in the midst of a crisis, a human-caused crisis, which is global warming. And we're going to talk about what are the consequences of some of this for the fungi, because all microbes will have to adapt or die. So global warming will have tremendous effects on the microbial world. I want to take you back 250 million years to the Permian. This was the greatest extinction event in the history of the biosphere. 90% of life on Earth died. And our current understanding is that it was a coal fire in what is now Siberia that dumped an enormous amount of CO2 into the atmosphere. That phenomenon happened over 80,000 years, with dumping the same amount of carbon over a couple of centuries. I think you know where I'm going, but I think it's worthwhile to read about the Permian to understand what may be ahead. And here is a new idea I want you to mull over. Whenever the Earth does poorly, the fungi do well. Here is a fungal spike of about a meter that separates the late Permian from the early Triassic. This is a mountain in South Africa. Think about it, 1 meter of fungal spores that are 250 million years old. In that devastated ecosystem of death and destruction, the fungi proliferated to enormous numbers. And those fungi resemble modern-day pathogenic fungi. Why am I telling you this? Because it means that all survivors would have had to deal with a lot of fungal diseases. And one of the things that we need to begin to think about is the impact of these filters that could have on the survivors. So the fungal kingdom today is enormous. It's probably the largest kingdom. It includes the major pathogens of plants, insects, and vertebrates. And currently, they're devastating various ecosystems. I'm about to tell you. But here is the remarkable thing. We don't worry about fungal diseases. Except maybe toenails, athlete's foot, but you don't expect them to kill you. We worry about bacterial diseases, viruses, malaria, parasites. Why don't we worry about the fungi? If the fungi are so destructive to plants, insects, invertebrates. There are, in fact, very few pathogenic human fungal species that are pathogenic for humans. So you have this enormous diversity out there, but very few have the capacity to cause disease. So I ask you the question, what makes us so resistant? Why mammals? Why are we so special? The origin of mammals actually goes back to the Permian. They were back there and they made it through that calamity. And mammals are different in that we maintain an internal stable environment. We maintain the same temperature from birth to death, except maybe a fever here and there. But for most of the past, the mammals were really not going anywhere. And part of the problem is that mammals require a tremendous amount of food. I already had 3 meals today, and when I finish here today, I'm going to have a 4th one. This is a huge amount. If I was a amphibian or a crocodile, I could only have to eat once a week. Here's a sense of how much energy mammals need. This is a log plot, and you can see that mammals need about 1,000 times more energy to get through the day than a frog, and 10,000 times more energy to get through than a fly. And mammals were going nowhere until the Cretaceous Paleogene event. And then you have the great mammalian radiation that gives us everything from whales to bats to humans. What is the evidence that endothermy or high temperatures protect against fungal diseases? Well, this is work that we've been doing in Baltimore, collecting fungi. You collect fungi from the environment, you take them into the lab, and you plate them. They are very happy at 30 degrees, but they don't do well at 37 degrees. Only one group, and this isolate happened to be a pathogenic isolate of the type that causes disease at 42 degrees. So today, when you look at what the fungi are wrecking, they're wrecking organisms that are cold-blooded. The frogs, the crops, the salamanders, the turtles, wheat, the snakes in North America. It is the cold-blooded animals that are at high risk for fungal diseases. Frogs can be saved. We are living through a time of tremendous amphibian declines caused by a single fungus. But if you create a frog sauna, this is a paper in Nature last year, just heat the heat blocks, the frogs know to climb on it, and they raise their body temperatures, and that is enough to cure the infection. Fungal diseases in humans are more common in the colder areas. The nails, athlete's foot, dandruff, by the way, is a fungal infection. The skin, and in babies, diaper rash caused by candida, the cooler parts of the skin. So people have looked at this in the lab, and this is the work of John Perfect at Duke, but basically, you take a bunny. The bunny has a temperature of 40 to 41 degrees, and you try to infect it with a pathogenic fungus known as Cryptococcus informis. You cannot give it a fungal infection. However, you could give it a fungal infection in the colder areas, like the testes and the skin. But if you give steroids for immunocompromised, temperature is not enough, and you have a dead bunny. This all put it together suggests that the reason that we don't worry about fungal diseases is because we are warm and we have adaptive immunity, and that gives us tremendous resistance. An example of this is the catastrophe that has happened to bats in North America. Bats in North America were doing well till 2006, and then a fungus was introduced from Europe. This fungus only grows at low temperatures. When the bats hibernate to get through the winter, the fungus grows on them and can kill them. If you take the bats into the laboratory and feed them, just raising the temperature is sufficient to clear the infection. Some years ago, we looked at the susceptibility of the fungi to temperature. We looked at 4,000 species, that's the study at the top, and we found that the great majority of them do well up to about 30 degrees. But then, as you go above 30 degrees, there is a sharp drop in their ability to grow. Remember, they are hypothermic. They love the cold. And My friend Aviv Bergman, who's a mathematician, took that data from the top, and as well as a well-known equation of how much energy you need to be warm, and he solved it simultaneously and asked, what is the optimal temperature by which you don't have to eat all the time and get the most protection against fungi? And the temperature was your temperature, 37 degrees. Now, that could be coincidence, but I don't think so. I'm going to argue to you that the fungi were critical in who we became as a species. And let's go back to the mammals. The mammals were back in the Permian. They spent the Jurassic and the Cretaceous not doing well. And then you have this event, which is known as the KT event, which was when a rock from outer space hit the planet. And you can see the dinosaurs go away, and now it's the age of mammals. It's the age of birds, which are warm too. The crocodiles make it through this calamity. So what killed the dinosaurs? Well, depends who you ask. People used to think that it was small mammals that ate the eggs. That doesn't make any sense. This doesn't make any sense either, but I find it funny. I don't think that smoking had anything to do with that. Now, there was a Deccan event about 65 million years ago. Those are the Deccan traps. They erupted. They're in India now, and they probably did a major job on the climate. So it's possible that major climatic changes contributed to their demise. But most people today think that the lethal hit came from an asteroid that hit Yucatan. And there are two other ideas that I'm going to share with you. One of them was published in a very respectable journal, and I leave it to whatever you think, but somebody actually proposed that they passed enough gas to create a climate change. I don't think this is right. And the next explanation I'm going to present to you with silence, because as a scientist, I have no data for or against it. So let's go to the Cretaceous world. It was a warm, volcanic, and forested planet. The Atlantic was tiny, and the world had this reptilian megafauna. And this asteroid hits the Yucatan, and you can still go there. The cenotes are at the rim, which are these holes that go into water very deep at the rim of the crater. There was massive deforestation. The blast was enormous. There were giant tsunamis swept through North America. And this is documented in the fossil record that the entire forest were wiped out. Photosynthesis gets shut down for at least 6 months, maybe as much as 10 years, because the fire, the dust, blocks out the sun and global temperatures drop. The earth becomes a giant compost, and there is evidence for that too. So in that decaying vegetation, the fungi thrive again, just like they did at the end of the Permian. And this gives you a sense of that. You had what you see there is the KT boundary, and then the iridium is the concentration is something that was brought in by the asteroids. And what you see is it's a world of conifers and flowering plants, and then it all dies. And you have the fungi, and eventually the ferns return. And in the early Paleocene, but think about it, anything that survives would have had to deal with massive number of fungal spores. The post-impact world, the reptilian fauna is probably killed by the blast, the disruption of food sources, the chilling of the planet. Reptiles fight off fungal infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases. The fungi can go in from the outside and go through the pores. In fact, we have fossil evidence that dinosaur eggs were susceptible to fungi. And this, a few years ago, I proposed that this fungal bloom at the end of the Cretaceous creates a filter that selects for the emergence of mammals. So this is a very simple idea. You have a calamity, and you have survivors. You have iguanas today, and you have mammals today. You have small mammals and small reptiles. But then there was a fungal bloom, and the fungal bloom could have helped keep down the reptiles. So when the sky is clear and the planet recovers, there are just a lot more small mammals. So you get larger founder populations, and we have the age of mammals, and we're all here in a Zoom webinar. So I've written about this. I wrote the first thing 20 years ago, and I update it every now and then, depending as new information becomes available. So this is a lot of explanatory power. It provides a selection mechanism for our energetically costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It argues that your temperature is a major host defense mechanism. And it also explains the minor role for fever today. Why? Because most of the infections that we get from other hosts, and the microbes are already adapted to temperature. Example, the SARS-CoV-2 virus, you get it from another person. But if you go out there on the Delaware Valley and you fall and you scrape yourself on a rock and you get dirt on it and you get a fever, those organisms are not adapted to our temperature. And fever can provide a tremendous protection. But we have a problem. The fungi can adapt to higher temperatures. So even though they love to cool, and even though they do very well in cool and dark environments, you can adapt them in the lab rapidly to grow at 37 degrees. And this was demonstrated by that paper on the right, where a company wanted to make a fungus to be a better control of insects. Insects get infected with this fungus, but they find a hot rock and they sun themselves. So they cure themselves. So they were able to train this insect fungus to grow at 37 degrees in a matter of weeks by simply slowly raising the temperature. And we have found evidence that when you look at collections, that is where people deposit isolates, if you compare the isolates over the years, this is noisy data, but I'm just going to give you the result. The paper is down there. Is the trends are upwards. It's the fungi began adapting in the mid-1980s, such that the isolates that we are banking today are more thermally resistant than the isolates of decades ago. The fungi are adapting to temperature. And in fact, we decided to look this in the city of Baltimore. Cities are heat islands. That is, they're often 4 or 5 degrees hotter than the temperature in the rural areas. And people have already looked at this, and they, for example, this was an early paper in which they compared fungal species from the city and fungal species from the rural areas. And the fungal species from the cities were already more temperature adapted. They could grow better at higher temperatures, telling you that fungal adaptation was happening in the cities. So we decided to look into Baltimore. Baltimore can get pretty hot in the summer. These are the number of hot days in downtown Baltimore City. It just goes up every year. And what we decided to do is to look at 2 neighborhoods. One that is cooler. You can see the green on that heat map of Baltimore. And another one that is much hotter. It's actually, that arrow is not right. It's that band of red. That's where we took it from, Fayette Street in Baltimore. This was done by Daniel Smith. And we asked the question, are the urban fungi more temperature resistant than expected? Are the fungi from warmer neighborhoods more thermotolerant than those from colder neighborhoods? And what are the differences? So the first thing that was amazing is that the fungi from the hotter neighborhoods are losing their pigments. Remember, melanin captures heat. So if you go to the warmer neighborhood, they're whiter. Their mean gray scale is higher. And that is, they're adapting and they're trying to stay cool by losing the pigments. But the most worrisome result was that we looked at some fungi that are pathogens. And when we took them into the lab and challenged them with high temperatures, the ones from the warmer neighborhood were about 3 times more heat resistant than the ones from the colder neighborhood. So within a city, within a distance of 4 miles, you are already having fungal adaptation to temperature. And then This is a paper that I published about more than 10 years ago, in which argued that global warming will bring new fungal diseases. Remember, you are protected against the fungi by your temperature. Most fungi cannot live in your body temperatures. But what if they adapt? They can then defeat your thermal resistance. And then, this is a headline from the New York Times in 2019 of a new mysterious fungal disease. And that's known as Candida auris. This is an organism that was not known to medicine until 2007. It was isolated from the ear canal of a patient. That's why the name auris in Latin. And retrospective analysis showed that it wasn't there. Maybe an occasional isolate was recovered. But here is the thing that is very difficult to explain. Then between 2009 and 2013, you had simultaneous emergencies in multiple continents. There have now been 6 emergencies, and the isolates are not related. It's not like somebody took a plane from South Africa to India. These things are coming out of the ground and coming. The question is, how did it emerge independently in 3 continents? Well, there are 3 theories. One of them is it was all there. It was always there, but people miss it. The problem is that has been ruled out. People have gone back to fungal collection. It was not there. It was the result of use of azoles in medicine and agriculture. Problem is, it's unclear how antibiotic selection will lead to new virulent species. And what we propose is that it's climate change. And the problem is, again, how do you prove something like this? Well, you can't. The only evidence that you can get is circumstantial. So we began to look at this. And the first thing you look at the relatives. Are the relatives more temperature resistant? And the answer is no. This organism appears to have adapted and acquired the ability to grow at higher temperatures relatively recently. And now it's been isolated in multiple places in India and in the Andaman Islands. And the organisms in the Andaman Islands, it was isolated in 2 places, from a beach that was frequented by people and from a wild marsh that people don't go into. The isolates from the wild marsh had struggled to grow at 37 degrees. It is almost that they're adapting, but they're not quite there. Now, the fungi are adapting, and now we have another problem. People are getting colder. How can that be? It turns out that you're colder than your great-grandparents. Well, this is a fascinating study where you looked at the temperature over many decades. And around 1900, people were warmer than we are today. Why? Because there was a lot of inflammatory diseases. People were infected with tuberculosis, worms, things like that. Inflammation makes heat. As we live in cleaner environments, we get less infectious diseases, we have less inflammation, and we're cooler. And global warming brings another problem. Whenever you raise the temperature, mutagenesis increases. Most of the mutations are deleterious, but occasional mutations give the fungus drug resistance and higher virulence. So this is happening all over the planet. Again, bringing in the concern that we are going to not only getting adaptation, but we may end up with nastier organisms. And I end with this, The Last of Us. So I did not know anything about The Last of Us, but I began to get emails when this thing came out, people saying to me, "Hey, Arturo, did you advise them?" And I said, "What are you talking about? I don't know anything about it." So I went and looked at it, and it's a very popular show that involved fungi that have adapted to become zombies as a result of climate change. And all the reporters wanted to ask me the same question: "Doc, is this possible?" And my answer to all of them was, it is improbable, but not impossible. Improbable because we don't know of any fungus that can turn people into zombies. But fungi can turn ants into zombies. Ants are animals. They can get into an ant, they can replace an immune system, their nervous system, they can make it climb up a tree, and then they kill it on the top of the tree. The ant becomes a fungal sac, and all the fungi drop down to the ground to infect new ants. And I also say to them, think about the following. LSD, where LSD comes from, where hallucinogenic mushrooms come from. Certain mushrooms have the capacity to give people altered states. So this is why I said it's not impossible. Is there a fungal species out there that if you get infected with will turn you into a zombie? I don't think so, but I can't rule it out. And with that, I thank you very much, and I hopefully stay within time. I think I did. I was told to finish by quarter off,
You're doing great.
and I
So,
did it.
fabulous presentation. There's so much going on there. We do have a few questions, and I invite others to put their questions in here. So, our first one is from Christian. Are there any kinds of fungi that give off a warm heat signature throughout the whole plant, or is it just around the edges?
That's a great question. I think the only answer I can give you is I don't know. I think that it would be very interesting to take the thermal camera and go find plants that are infected with fungi and take pictures and see if there is a difference in temperature. We haven't done it. If we have time, I think it's a great idea. Just got to find the right tree, the right place to take a picture.
Cool. Christian had another question too. Since fungi are growing on the space, and since they thrive in areas of high radiation, do they not need oxygen to grow like other plants?
That is an excellent question. The fungi tend to be aerobic. So I don't know if they can grow in the vacuum of space, but we do know that if you expose them to the vacuum of space, people have done this already and they can hang out for a year in the cold of space with all that radiation. So they may not be able to grow, but they can certainly survive. And that raises ideas of, the danger that we have when we send probes to other planets. We may be putting organisms there, as well as that has to do with the idea of how did life emerge in this planet. There is the panspermia hypothesis that life was seeded in this planet from elsewhere, and this accounts from the fact that we all have DNA, RNA, that we all came from one nidus. But it's a big question.
Anonymous asks, very interesting idea about the fungal bloom contributing to the rise of mammals. Have you heard recent proposals that dinosaurs were endothermic or at least had some control to elevate body temperature? Has this caused any feedback for your fungi rise of mammals idea?
Yeah. So this is a frequent question that I get, and if I, I don't think calamity. They are at the top of the food chain. The planet is devastated. The planet temperature drops 10 degrees. I think that they die from the calamity. What I'm interested in is if you had surviving reptiles and surviving mammals, how do we end up with the age of mammals when you're dealing with organisms that have to eat so much?
Yeah.
And that is, people say, well, the dinosaur died, so it created a space for the mammals. I said, really? What does that mean? Because there is also more space for the small reptiles.
Yeah. And then Michelle asks, fascinating information. If you could wave a magic wand, what should we do on a personal level and a global level to start getting ready to combat the future changes or dangers of fungi?
So the second part I think, is easier. So what I think we need to do is we need to map the natural world. We need to basically embrace the One Health concept. We don't know what's out there. I mean, people don't do this kind of work to go out there into the Delaware Valley and to map the fungal fauna. We don't know. What little we know from collectors, people are interested in mycology and things like that. So we need to invest to know the natural world. This is not rocket science research, but this is very necessary so we know what's out there. And I think that will bring tremendous dividends. We're going to learn about new species. We may have new drugs. We may have things that we can't even conceive right now. At a personal level, I think that we are all scientists and we all need to fight for science. And I think that that is very important. And the science from the very beginning has always been under attack. You go back, the Galileo trials, the Lysenko situation in the Soviet Union. Once science begins to affect an element of society, it gets pushed back. Let me give you an example. Today, there is no anti-science movement against quantum mechanics, right? You don't have protests in the street. We hate quantum mechanics. Tell us what the position of the electron is. I don't believe you. Why? Because it doesn't really affect some aspects of society. But once it does, you could easily have it. And what we need to say is we need to basically look at the big picture. Science is humanity's greatest invention, and we need to protect it. And if you don't think so, what time in the past would you like to go to? The world of your great grandparents, where 1 out of 3 children didn't make it to 5. It's given us the world that we have, and I think that if we are smart, that we can navigate this crisis, and that we can even come out ahead. But we all need to become foot soldiers for this enormous enterprise that is giving us so much good.
That's an excellent statement. I just reminded me talking about, that we all can go out and learn more about fungi. I've got a colleague of mine. I've been a member of the Pennsylvania Biological Survey for quite some time, and I'm a mammologist, so obviously I'm more interested in mammals. But he's a mycologist, and it's been a constant fight within even that group. We have orphan taxa, and fungi are certainly among that orphan taxa that nobody is paying any attention to. Agencies are not paying attention to it.
I know.
Are you seeing any growth in science community that wants to?
Yes. Yeah, and I've been involved with someone. There is a large room for citizen science. Like, for example, now when mycologists go out into the woods and take a picture, you can geolocate it.
Mhm.
You can get a lot of information. I give you a simple thing. I bought my wife a bird feeder with a picture, with a camera. So every day now we're looking at what birds come in.
Yeah.
That you can imagine that if you had a few hundred bird seeders in Baltimore like this, we could get a pretty good census
Mhm.
for what's going on out there. So I think there is a tremendous need for citizen science because the technologies allows us to collect a lot of information. And this information can be deposited in places, in mycology, for example, communities like that, and interest groups that to follow a lot of this. And so I think all of you should look around you and say, what can I contribute? As well as make sure that you hold up the flag of science.
We got one other question here from Anonymous. Do pharmaceutical companies have a plan for antifungal medications with the recent rising of fungal pathogens? Is there anything that could be done to improve these antibiotics?
Yeah, our best antifungal was made on the year that I was born. Okay, and you take a look at me, and I've been around for a while. Okay, the companies are trying to develop this company, and we have new antifungals coming in, but the problem fundamentally is that it is hard to find them, and because the number of fungal diseases is relatively small, they often don't get as much investment. The problem with fungal diseases is that they are often a low probability, but very high consequence event. That is, people who are immunosuppressed who get a fungal disease, the mortality is just very, very high. So the pharmaceutical company is trying, new drugs are coming in, but again, the road is hard. The road is hard.
And being in mammology, I just like to end a little bit about white nose syndrome. And so, that was a devastating event for mammalogists in America when we started doing normal surveys of hibernacula and finding thousands and thousands of bats dead. A large number of species with declines that are 95 to 99% due to this fungus. And then the problem that, what do you do about it? It's relatively easy to take care of it on an individual bat. But when you have a hibernaculum, the bats leave in the spring, the ones that have survived will survive after that, but they can get infected the next year from a single spore that's been sitting there waiting for them to come back. Um, this is really a struggle that's
So I
for us.
encourage, so people have tried all kinds of things. They try to dry out the caves, which is impossible. They try to raise the temperature in the caves. Uh, but there's a group in Wisconsin with my friend Bruce Klein. They have developed a vaccine. And
Really?
yeah, and you can find it. They had a paper earlier this year, and the idea is to get a vaccine that you could smear in at least a few of them. They're very
Oh, yeah.
social, so they smear each other and
Mm.
vaccinate the whole cave that way.
Well, one of the things that Pennsylvania's been doing is that they recognize that the fungus has a relatively narrow range of preference. And so what they've been doing is actually trying to modify former mines into making them much colder. The
Interesting.
bats, bats like it, but the fungus doesn't
Doesn't so it
doesn't
basically
impact them.
it gives them a protected heaven. I mean, look, a lot of the news is depressing, but I leave you with one thought. Here is one species trying to really help another species. And that a lot of what you read in the news is very depressing, but that is something that gives me a lot of hope and encouragement.
Well, I thank you for that. It's always great to kind of end with some hope and some encouragement. Dr. Casadevall, I really appreciate you being with us here this evening. I thank all of you who joined us for being here this evening, but keep in mind, if you missed some of the items or you want to go back and see it again, it will be posted on our website very soon, probably tomorrow morning. So you'll be able to come back and catch up on some of the things you might have missed. Thank you so much. I hope you
Thank you
have a
for having
great
me, and good night, everybody.
good night, everybody. Take care.